Hello, everyone. Thank you for joining in. I'm Mahesh Bashyal, a PhD candidate in the agronomy department at University of Florida. And today I'm going to talk about our current research project that is ongoing in our research station, which is entitled Do Below Ground Peanut Plant Parts Contribute and Credit to Subsequent Crops? And let's start with the rationale. And as you know, the extension recommendations behind peanut cultivation recommends around 22 to 67 kg nitrogen credit after peanut, but these have not been substantiated by the scientific literature. Especially if we look into the literature, we find that field decomposition litter bag studies are often used to estimate estimate N credit from peanut residues to subsequent crops. However, the problem is the scientific literature has never taken into account the below ground plant parts and undug pots, and most of the litter bag studies, they are only focused on above ground plant residues, that is leaf and stem, as discussed by recent papers by Johnny et al. 2020 and other papers by Mulvaney et al. So the idea of this research project is to quantify below ground N contribution from peanut residues, especially undug pods and roots, and inform peanut sustainability initiative by quantifying N availability from these residues to subsequent crops, and quantify fertilizer replacement value related to these practices. So the hypothesis of our study is peanut roots with nodules and undug pods contribute a significant quantity of N to subsequent crops. And the objective for our study, of course, is to quantify these contributions of peanut plant parts, especially undug pods and roots, to N availability for synchronicity with subsequent crops. Starting with the experimental design, this was a randomized complete block design with 4 replications conducted during 2020-2021 at Jay, Florida. And we had 4 treatments as 4 different plant parts: leaves, stems, roots, and pods. Actually, we partitioned peanut plant parts into these pods, roots, stems, and leaves. And residue loads for this study were based on average total peanut biomass, which included total biomass included above ground and below ground biomass in Southeast US conditions, and plant parts were partitioned proportional to the total biomass and placed in litter bags. And the litter bags were retrieved at 0, which was the time 0, 3.5, 7, 14, 28, 56, 112, 222, 4, and 365 days after deployment. Actually, we are in this phase of the litter bag retrieval at this time, and we are still waiting to process and analyze data from this time point and this time point. So talking about the materials and methods, let's start with the data analysis. We are analyzing data with proteomics procedure and SAS, especially for the mean separation or analysis of variance at 95% confidence level using least significant difference. And we plan to use a proc core to estimate correlations, especially with the initial fiber quality with the mass loss, which I'm going to talk later. And least square estimates for nonlinear models were determined using exponential decay models through Sigma plot software from Systat Incorporations. And this is an experimental plan. This I included this to explain to you what is the schematic calculation for estimating N credits to subsequent crops. And this is from one of my extension paper from my advisor, Dr. Mike Mulvaney. And as you can see, when we are trying to find the N credit, which would theoretically equal to the difference in N remaining from the time of litter bag deployment to the time of wheat harvest. For example, we deploy litter bag here, and let's say after a few days, we plant winter wheat. And then when we harvest winter wheat around this period, the difference between the N remaining at this point and at this point would equal to the N credit available during the winter wheat growing period. So this is the schematic plan or idea that we use to calculate it and available to subsequent crops for this study. And let's talk about soil moisture and temperature and rainfall first. As you know, soil moisture and temperature data are essential in decomposition studies, as the mineralization process is dependent upon the microbial activity. And this could be influenced by moisture and temperature. And the data here, it makes sense. And as you can see, this is the tensiometric soil moisture data, where higher values denote drought conditions, whereas values close to zero indicate soil moisture at fully saturated condition, and we have soil temperature at this axis. And this data makes sense because, as you can notice, after a heavy rainfall, which is indicated in this gray line, there is a decrease in soil moisture KPA, which indicates higher moisture availability, and the temperature also decreases after the rainfall event. So overall, the data makes sense, and we decided to characterize the rainfall because this helps to rainfall and temperature, as I explained before, this helps to explain the process of mineralization, the process during the experiment. And we also included an initial fiber quality analysis of the peanut plant parts. And literature, the question arises, why are we including this figure? And literature has shown that in order for residues to mineralize faster, and concentration of the residue should be higher, CN ratio should be lower, lignin to nitrogen ratio should be lower, and ADF to N ratio should be lower. So that's why it's important to include fiber quality in our analysis, especially what we plan to do is we plan to correlate the mass or N loss remaining at a certain point in time with this initial fiber quality to see if there is any correlation or not. And we would expect the residue with our different peanut plant parts with lower lignin to N ratio, lower ADF to N ratio, and lower CN ratio would have higher mineralization. So now let's talk about the biomass mineralization from peanut residues. And before we delve down into this diagram, I just wanted to touch base first on this decay equations here. And as you can see, leaf and stem mass loss were best described by a 4-parameter double exponential decay equation. And as you can see, this denotes the labile portion of the residue. This denotes the recalcitrant portion of the residues. And these are the rate of decay for the labile as well as for the recalcitrant part. And the greater values indicate higher mineralization. And as you can see, double exponential 4-parameter models were good fit to describe leaf and stem mass loss, whereas for roots, 3-parameter signal exponential model was a good fit, whereas for pod, 2-parameter signal exponential model was a good fit. Similarly, to describe the carbon loss or C remaining during our experiment, we noticed that leaf and stem, as similar to biomass loss, were best described by 4-parameter double exponential decay equation, whereas roots and pods were described by 3-parameter single exponential and 2-parameter single exponential models respectively. And as you can notice, buried carbon loss model appeared similar to buried mass loss model because most mass loss was carried due to microbial respiration of carbon, which was then lost probably to the environment as CO2. So you can notice that. Now let's talk about the nitrogen mineralization from peanut residues. And here we can observe that there is an immobilization from stem residues compared to the leaf and root decomposition, because stem residue tends to have higher CN ratio. And we notice usually when the values are greater than the initial values, then there is an N immobilization in the system, which indicates that nitrogen is entering the system rather than N is being mineralized. So we can also notice that peanut pod decomposes at a very slow rate, which was similar to carbon and mass loss. So now coming to the big picture here, why are we using all these equation models? So our overall objective is in coming back to that original figure where I showed a slide of where we used any 2 time points to calculate the difference. to find the N mineralization to subsequent crops. I did the C and N mineralize during a hypothetical wheat production, which was 112 days after deployment. And as you can see, when I subtracted the nitrogen concentration, nitrogen content remaining at 2 different time points, as you can see, we have roots and undug roots and pods contributing small but important and contribution to subsequent crops. So this takes back us to our original question, that do we see nitrogen contribution from peanut residues and undug pods? And from our preliminary data, and we still have 2 more time points to collect for this season, and we are planning to, of course, repeat this trial. We see that from our preliminary conclusions are a small but potential significant contribution of peanut roots and pods for subsequent crops, nutrient synchronicity. And traditional decomposition studies utilizing above ground residues only may underestimate N credit. So I think we need to look into this direction for the future as well. So with that, I would like to acknowledge the references that I used for my studies. I would like to acknowledge National Peanut Board for funding this project, our lab crew for helping the partitioning and the litterbag deployments and all our work, and our technician. And with that, I'm open to questions. Thank you.
Thank you very much, Mahesh. Nice presentation. We do have a couple questions. First question is from Liang. Could you please explain why soil moisture drops after rainfall? Thank you.
Okay. So if I understand the question correctly, Leon's question is, why does the soil moisture drop after rainfall? So Leon, to answer your question, so this is a tensiometric soil moisture sensor. And usually in this kind of system, we are trying to emulate the roots. For example, the higher values indicate the root has to exert more pressure in the soil to suck the water out into the roots, right? So the pressure of kilopascals goes high on the dry condition, as you can see here. But after the event of rainfall, which we can see in the spike in this gray area, so the water is kind of saturated here. So plant has to exert less pressure. So the tensiometric reading is low. So this is actually a tensiometric reading which decreases after the event of the rainfall. Thank you.
Okay. Question I have is, based upon your results, if you were to speak to growers in the panhandle of Florida, what's your recommendations for, as far as based upon this, what is the nitrogen credits or the carbon credits? How would you advise growers going into planting wheat, how they might adjust their fertilization plans?
That's a great question, Dr. Kimmerer. Thank you for that. So Dr. Kimmerer's question is, what would be my recommendation to growers based on the preliminary results that I have in terms of like nitrogen credit, if we have to give the nitrogen credit to the subsequent crop or not? I would say, Dr. Kimmerer, based on what we have seen so far, we see small amount of nitrogen to subsequent crops, but and if we take into account the roots and the pods, which was the main objective and which we have included in our research, we could say, maybe we ought to give a little bit more N credit. But from our data, extensive data collection that we have seen when we have taken yield data, when we have taken yield data for the subsequent crop, we do not see N credit, especially for the panhandle situation. It could be that there is nitrogen leaching, and this, especially one of the caveat of our studies, we didn't discuss the N fate, and this study does not discuss about the N fate, and especially in sandy soils and Florida condition, I think it's very hard to retain nitrogen for subsequent crops. So I wouldn't necessarily recommend to reduce N credit, especially based on how the weather is here, faster decomposition, lower N availability, low CEC soils, and yeah, and based on our yield data, which shows that maybe it's due to an immobilization from cotton residues. I didn't talk about cotton residues here, but when we compare peanut versus like cotton in a cropping system, and what we have seen is like cotton residues might contribute to immobilization, and when we compare with the peanut, it might look like we are getting a peanut end credit, but when we compare with the true fallow, the fallow in peanut, especially the yield response behind fallow and peanut, tend to behave similarly. So I won't, long story short, Dr. Kimbarat, I won't necessarily recommend growers to reduce end credit, to reduce end fertilization for the panhandle.
Thank you. Very, very thorough answer on that. And that's the time we have. Thank you very much, Mahesh, for participating in this JOSUG session, and thanks for your presentation.
Thank you, Dr. Kimbarat.